What I see is that a lot of people are trying to run the pipelines. I think it's very important that we all incorporate the online materials that we discussed, because we're going to be talking briefly about gene expression today, for example. So I'll talk a little bit about bioinformatics or transcriptomic data analysis, RNA-seq. If you're not familiar with that, we have a course on RNA-seq, a collection of courses. We also have several projects that could be beneficial. So we have a couple of things in the forum that I'll briefly address. I posted the tools and the links to the tools that we discussed last time. I don't know if any one of you has been able to install some of those tools. If you have, please let me know. I will be using some of those tools today. We talked about having a way to visualize structures of proteins. We talked about building phylogenetic trees and annotating some of those outcomes from the pipelines that we've run. There have been some publications that were mentioned there as well. And some of you were asking last time, how do we create certain charts, right? So we're gonna do that today. And there was another question. All right, great. So Eugene, Fig Tree, great. We'll use both of those today. And also, hopefully you had a chance to install Chimera or Chimera X. I think also very useful. Okay, so some of the questions about the browser. So we tried looking into the problems that some people had with the browser. The best browser to use is Chrome. Okay, somebody had challenges with Fig Tree. I don't know what that might be. We had, so let's go to the next page. We also discussed how we can process data. So there was a question here. If a data file is clean, does this mean we do not need to perform pre-processing? And what is the seed substring for bowtie to align? Does bowtie to align map the reads to all the viral genome elements from NCBI? What is the difference between reads being paired and aligned concordantly? So some of these are great questions related to probably mapping of the reads on the reference genome or the database. So I'll talk a little bit about that actually in our one of our future sessions, but I will respond to this separately on the forum. Some other people were trying to run some of the previous pipelines, and I've interacted with you to share with you some of the files. But we will also maybe create a separate forum topic for all of the files. And so I just want to make sure that everybody sees some of these things that were posted since last time. Here we have session data files and presentations and tools and resources. Okay, I'm not sure what might be the problem downloading Fig Tree, but if you were able to install Eugene, I think that probably has that as well. Okay, so here are some links to some of these tools. Fig Tree, I'm not sure what might be the problem, but if you have a specific error that you have encountered, we can look into that. Also, we will talk a little bit about this, but if you want to create some of these charts, I posted here, some Windows programs. I don't know how many people are using Windows, but here's some Windows programs. And if you're familiar with R or Bioconductor, you can use this neat program that generates some interesting charts. And then I'll just show what everybody I think can do is something in Microsoft Excel. It also builds up the logic for those that want to understand where these figures come from. All right, so some good discussions, and hopefully everybody is taking advantage of that. So let's briefly review what we did last time. Last time we talked about viral adaptation between different hosts. So we talked about bats, we talked about humans, we talked about some of the ideas about the origins of viral outbreaks and how viral transmission happens and what actually happens in different tissues. Specifically, we talked about the proteins that are responsible for cell entry, and those proteins are not expressed everywhere, not in all tissues. And so the virus is obviously specific to a tissue. That kind of also determines the transmission being airborne, the transmission between the fecal oral route, the transmission from other ways of transmission. All of these things are also determined, for example, through blood, right? So they're also determined by the specific cell types that the virus is effective at entering. It also has to do with viral load, so how much virus can enter into and in what type of entry that is. So we will talk more about that. We talked about different viral proteins, right? And we also looked at the Ebola virus glycoprotein. And the glycoprotein is actually just a whole type of viral proteins that are typically on the surface exposed, like we see here in the Ebola virus. You can see that the RNA is inside, tightly packaged, and then you have the membrane, and then you have this protein that sticks out. And so if you are interested in Ebola and have been following what's been happening with Ebola, of course, this is also the protein that was used to develop a vaccine. And so what was important is to identify specific mutations on the surface of this protein to make sure that the vaccine is effective. And we looked at how phylogenetically we can understand the relationships between the different viruses coming from bats, coming from different outbreaks throughout the years, and compare them. And so we're able to see that the glycoprotein is something that has a lot of significance for a lot of different viruses. Ebola virus, and then also we started talking about coronaviruses. And coronaviruses they also have a glycoprotein, a spike glycoprotein. In fact, that's why the coronavirus is called coronavirus, because of that glycoprotein. And hopefully all of you had a chance to start reading about that spike glycoprotein, which is something that we'll talk about more today. So then we reviewed this new section that tries to combine several different important types of analysis that are going to be useful for the types of things that we do, right? So what are those different things? We have an ability to map our sequence reads from next generation sequencing FASTQ files, align them to a genome, pre-process them, clean them. We have then the ability to identify point mutations and measure mutability, or how much does each position mutate in a specific sample or group of samples. We also have an ability to compare group to group, multiple samples in each group to determine significant mutations, and we'll talk more about that in later sections. Then we also can apply that to time course development. And here we have a whole section dedicated to defective viral genomes. So those are important. We'll see in different types of viruses, they play a major role in how the disease spreads and what kinds of symptoms it has. And the major idea there is to not look at point mutations, but look at whole sections of the genome that are missing. And so that is going to be important because, of course, we cannot accurately determine those just in a single read, especially if they're longer than a single read. So accurately understanding what the position of cutoff is important to be able to understand defective viral genomes. And then already a section that you're all familiar with, phylogenetic analysis, where we can look at haplotypes present in a NGS FASTQ files, so multiple consensus genomes. We can build a consensus genome. We can align those between themselves using nucleotide, amino acid sequences, or specific positions in the codon, which might have a significance from an evolutionary standpoint, and then build a phylogenetic tree using different methods, different models, different mathematical models that represent a process that we think is happening. And then we will talk more in the future how we can compare those different methods and understand what is a reliable and interpretable tree that we can then take further into interpretation. And so one of the things that we did last time was we built a pipeline to understand differences between these different coronaviruses. So I don't know if any of you had a chance to do that since last time, but I shared with you the files. Those files, again, just simply map between themselves into a multiple sequence alignment, and then we can either build a bunch of different trees or select one type of a tree. And then what we want to do briefly today is take a look at the pipeline output. So did anyone run this pipeline? All right, great. So did anyone look at their tree and understand kind of which sequences were similar to each other, and maybe some of the, okay, so somebody's pipeline is not finished. Yeah, some of these pipelines, they're in queue. So if a lot of people at the same time are running the same pipeline, you're just waiting in line, essentially. So some of them might take a little bit of time. But it's great that some of you did run it. I ran it as well. So I'm going to just show you my pipeline here as an example for everyone else that wants to take a look at what's going on. So let's go to the server. And I will use this. Let's take a look at some of these outputs here. Okay, and Nichiravan, please let me know what the problem is. So the best thing, again, if you have an issue as you're running through the pipelines or something like that, is to post it on the forum so we know in advance. We can address that in one of the sessions. But if you want to describe your problem here as well, I'll take a look at that too. So what are some of the things that we observe in the outputs? We have a FASTA output of the alignment, multiple sequence alignment. We have a PDF. We have actually a number of different FASTA outputs because they're going to be by group. So you can see this group 2, group 3, group 4, etc. And you also have the full amino acid FASTA alignments. You have this full table, so we'll take a look at that today as well. Different PDFs, etc. And there's also going to be a tree file. So make sure that you're actually taking the tree file from all of the alignments. So I will take this file and I'm going to open it in Figtree. Okay, and so this is what it looks like. So again, let me know if you were able to kind of do the same thing. Okay, so Nachirvan, if you can send a screenshot, I'd be happy to see what you are seeing so that we can address that. Okay, so what do we see here, right? So these are some of those input files. You can see group 1, group 2, group 3, group 4. You can see that obviously these groups are based on how you imported and put them into different groups. I used 4 groups, but I actually combined some of the different files that we had, like MERS and SARS-1, into one group. So what do we see here? Here we have, you can see group 1, but this is actually MERS, right? Here you can see group 3, and this is BAT SARS-1. Here you see group 1, and that is just SARS, human SARS. This is human MERS, and this is human SARS, so also group 1. And so we can see that the bat SARS-1 and the human SARS-1 are closer today, closer to each other. Okay, here we have all of the pangolin sequences, almost all of the pangolin sequences, so that's group 2. And then we also see these pangolin sequences right here in group 2. And then here we have a couple of bat sequences, so that's one of the COV2, C45, and this is RADG13. Right, and then here we actually have both a reference genome and some of those COVID-19. So I annotated this by looking at the different accession numbers and organizing the branches accordingly. You can do so as well. And so what we see here is we can add rate. This is rate of mutation. And you can see how rate of mutation actually helps us understand the closeness, the relatedness between these different sequences, right? So what does it essentially mean? Essentially it means that we have these branches, these nodes in the tree that are similar to each other. And you can see that the pangolin and the RADG13 are the closest to SARS-CoV-2, right? So hopefully that's similar to what you got. What is another, so, you know, some things to remember, you have some settings here. We're always going to be using the last tree because this is an iterative procedure. So we're going to be using the last tree, and you can play around with some of the labeling and coloring based on the rate of mutations and based on whatever selection you are going to make. Okay, so hopefully this is similar to what everybody else got. Now then, I mentioned how we have this multiple sequence alignment in amino acids, right? So we already talked a lot about nucleotide sequences. Now let's take a look at some of those sequences on the amino acid level. So here, this is in Eugene. Again, you can see that I added this information here in the groups. And so we have COVID-19, SARS, bat SARS-1, pangolin, bat SARS-1, pangolin, and MERS, right? So we have all of these different sequences, and we can actually see, right? So if we just look over down here, we see a lot of missing, a lot of changes, right? So the alignment actually shows us that not all sequences were created equal. Some of them have a lot of missing information. Some of them might have a lot of changes. And this doesn't provide us with an overview in general. This just provides us with every single position. What are the changes that we observe there? And we can kind of use the different coloring techniques and the consensus to view a dissimilarity or similarity between these different sequences. Now, the other file that we looked at, so I'm going to close the fig tree for now. Okay, the other file that we said was an output from last time was actually the TSV that has all of the sequences together, right? And so it looked like this. It was in TSV format, but essentially this is something that we can open in Excel. So let me show you what we can understand from that Excel file or from that table in terms of sequence similarity, and kind of link it to what we reviewed last time in some of the papers that we had. Okay, so essentially our goal is to create this graphic. This is a chart that shows percentage similarity by nucleotide. Of course, you can also determine whether you want to do it by nucleotide or by amino acid, but this is to build our understanding about how these types of charts are generated and what do they essentially mean. So we have in the original output something that looks like this. We have the position on the reference genome. We have here the reference genome that was used for framing of codons. Then we will have the amino acid sequence. So this is going to be the nucleotide sequence. This is going to be the amino acid sequence, and so forth for each one of our genomes, right? Because this is a virus, it's not going to have the negative strand, it's not going to have the alternatives. So we're not going to have any of the other ones. So we can just take, if we're interested in nucleotides, we can take just the nucleotide sequence. So what I did here was exactly that. I took the position, reference, nucleotide sequence, and then for each one of the nucleotide sequences, I organized them by group. So we have now SARS-1, bat SARS-1, COVID-19, and pangolin. And of course, not all of these are actually SARS-1. This is group 1, but some of these are MERS. Okay, so then what we can do, right, is do a simple calculation. And again, I'm doing this to illustrate to you how we think about the similarity. This is a little bit of manual work. It's not a lot, but I think it really helps you understand how do we get these numbers, right? Okay, so how would we calculate similarity for a group based on these types of sequences? Well, the idea is pretty straightforward. Let's say we have for all of them, right? So here you can see that all we are doing is calculating. So count if is a function to calculate if the if whatever cell value is equals to something in the reference, right? So I'm just going to use the reference, right? So I'm going to use this as the reference. And if all of these match the reference, I'm going to count them. If they don't match, I'm just not going to count them. So there's a total of 29 samples. And then I can divide whatever I got here by 29. So you can see as a result, 1 will be 100% similarity. And as these numbers change, that is how I get the percentage of similarity, okay? So then instead of doing this for all of them, I can do it separately for each one of the groups that I'm interested in. So for example, I'm interested in comparing pangolin, human COVID-19, and bat SARS-1. So now I can create a chart, right? So here's the chart. I have a COVID-19 similarity, and you can see that the similarity between all of the genome sequences that I had here to the reference genome is very high, right? So here you can see just a few changes from the reference genome. But then the different other ones, pangolin is blue, right? So right here I have pangolin is blue, COVID-19, bat SARS is pink, COVID-19 is orange, and overall similarity is red. And maybe we don't even need overall similarity because as we spoke last time, we're interested to understand how we think about the pangolin potentially being an intermediate host. And we'll talk a lot more about this in our next session, but for now, let me know what you think based on this chart might be the region where we observe a drop in similarity from COVID-19. What do you think? So again, the x-axis is the genome, position on genome. The y-axis is percentage similarity to the reference genome of COVID-19. Based on what you see here, the blue is pangolin, the pink is bat SARS-1 or bat SARS. What do you think might be the region where we see a lot of dissimilarity? What are some of those regions that you see here with a lot of dissimilarity? Okay, close to 3481, 3400. Okay, 22,000 to 25,000, right? So, okay, so this is around I guess what one of those, right? 3,400 or 3,600, right? This is that 22,000 to 25,000. Okay, great. So maybe also over here, right? This is 70% similarity. So we can just take that as a threshold and say, okay, these are some of those regions. Does anyone know what protein is in the beginning of the genome of the coronavirus? Has anyone had a chance to look? No, not glycoprotein. No, not S. In the beginning, ORF1AB, exactly, right. So ORF1AB is a polyprotein that is like the Swiss Army knife for the protein that it can use in multiple ways as it starts the replication process. Where is the spike glycoprotein? Who remembers where the spike protein is? 680. Well, right. So maybe you're not exactly. I think 680 is a good number to keep in mind for the future of this lecture, but that's not where the spike glycoprotein. Remember, this is nucleotide sequences and not amino acid, right? Exactly, Claudia, right, 22,000. So this right here is exactly the spike glycoprotein. Okay, so what do we see, right? Remember from last time, here we see essentially a small region, right? So this means that there's a specific change. In fact, we will talk about this later from the host response and how it changes when we see different mutations. We see that ORF1AB plays an important role in a lot of the disease progression phenotypes. But we'll leave it for now, right? We'll talk about the spike glycoprotein. What we talked about last time is that the accumulation of these different mutations overall, the dissimilarity overall, is important because we really see here how adaptation between different hosts is centered around this protein. And this makes a lot of sense, right? So we have a cell, the cell has a receptor, the receptors are going to be different between different species, and to adapt, to go between these hosts, the virus has to change. What you also see is that in this region, what would you say is the closer genome to the human SARS-CoV-2? Is it the bat or the pangolin in the spike glycoprotein? Okay, Martin thinks it's pangolin. Any other opinions? The colors are pink is bat, blue is pangolin, orange is human COVID-19. All right, great, pangolin. Exactly. So the pangolin, surprisingly, is closer to the human spike glycoprotein, which might give us some suggestion about how this might be considered as an intermediate host as opposed to a direct transfer from the bat, right? So at least we'll go in that direction. For now, we'll talk a lot more about how this works in real life. But if you look overall, right, overall they're fairly close. In a lot of areas, the bats are a little bit closer to the human sequence, right? So this is a similarity plot, important for other papers that you will see. And as we saw last time, in fact, exactly what we saw in some of those publications. So let's take a look at what we saw last time. Okay, so this is what we see, right? Where is this exactly? So these are the different genes. So this is the ORF1B, and this is the spike glycoprotein, starts right here. In fact, that's exactly, so based on this, does anyone know where in the spike glycoprotein do we see the most change? Or yeah, the most dissimilarity? Based on the reading of the paper that we did last time. I don't know if you had time to review that paper. So anyways, we'll talk about this more today. right? So the spike glycoprotein actually has two parts, the bottom, right? Exactly, the bottom and the top, the S1 and S2. And so this is the S1. So we'll talk a lot more about this part of the protein today. What did we see last time? So last time we saw exactly the same picture, right? So compare what we see, these two different dissimilarities, sorry, what we saw here. Again, we see it here. Notice that the y-axis has been cut off right around 30% perhaps. So we see a more pronounced effect right here. Okay, to generate these plots automatically, as I mentioned in the forum, there are a number of different tools that you can use, and here are some references to them. So if you would like to have a detailed view of each position and you want to generate some of these plots, our studio has this bioconductor library that has this specific package. I think is very useful, but also you have some Windows programs depending on what you prefer to use. Essentially you see that the results are going to be very similar, right? So we're going to look at similarity plots, and based on those similarity plots, we can understand relationships between groups of samples, just like what we did briefly here in Excel. Okay, so for the end of the last session, we had this assignment or a suggestion to read some paper to think about today's topic, which was based on this publication from, I believe, early 2020 called Structure, Function, and Antigenicity of the SARS-CoV-2 Spike Glycoprotein. We'll use this paper to think a little bit more in detail about this spike glycoprotein and what essentially does it do for the different types of disease symptom and progression, right, and outcomes. So how does this disease, which is a condition of the host, linked to what the virus does using these different tools that it has at its disposal encoded in the genome. So number one, state the similar mechanisms SARS-CoV and SARS-CoV-2 used to enter host cells. Has anyone read the paper? Has anyone had time to glance through the paper and some of the figures? All right, great. So do you remember, is there a similarity? Is there a lot of similarity between how, in general, SARS-1 and SARS-2 enter the cell? ACE2 receptor, okay, great. So, all right, so there is a lot of similarity at how they enter. All right, great. Number two, describe the structure of the transmembrane spike glycoprotein in coronaviruses. What are the general functions of the S1 and S2 domains? So who remembers, for example, which part of ACE2 actually connects, or sorry, which part of the glycoprotein, the spike glycoprotein, connects to S2? All right, great. So binding, right, receptor binding domain. All right, S2 helps fusion. Great. All right, so great. I see that you really read. The S-glycoprotein from SARS-CoV-2 contains a 4 amino acid insertion at position 681, 684. Somebody mentioned that 680 position, so that was great. What unique feature did this insertion create? Who remembers why that part was important? Furin cleavage, okay, great. Furin cleavage, yep, exactly. Okay, and so we'll talk a little bit more about that. And why is the viral S-glycoprotein a target of neutralizing antibodies in vaccine development? Blocked by antibodies, glycoproteins are good antigens, specific and blocks binding where ACE2 engages glycoprotein. Well, right, so one of the reasons is obviously it's exposed, right? It's actually the only part of the virus that is exposed without really having access to some of the more internal proteins. Nothing really is possible. So That's something that we'll talk about today. And antibodies, of course, have to have access. And the second thing is, if we're talking about preventing disease, right, we have to have something that happens before the virus actually enters a lot of cells. And so having that as the center of the antibody to block, right? So the antibodies essentially would connect to that spike glycoprotein, prevent it from getting into the cell. All right, great. So we have some background about what we want to discuss in this session. So we'll talk about, so we looked at similarity, right? The similarity actually could be determined in multiple ways. It could be determined by looking at different scores of similarity. It could be by looking at the amino acid sequence. It could be by looking at the nucleotide sequence. So there's a lot of variation to what you'll see, but the basic idea is always the same. If we see similarity, we can think about evolutionary relationship. If we see dissimilarity, that is also important because we can start seeing how the branch continues to separate and become farther away, right? So if we take this idea of similarity and we think about this glycoprotein, right, we can go back to our tree and the alignment, right? And think about, okay, well, what are some of these reasons for the differences that we see, right? And one of the things that we saw from the similarity plot is this section of the tree right here, right? And in the section, we saw how the pangolin sequences are close, right, between these bat COVID-19 or similar to COVID-19 SARS-CoV-2 sequences, and all of them are a lot closer to COVID-19 than bat SARS-1 and human SARS-1, and of course, a lot closer than MERS, right? So if we think of the origin, right? This is the origin right here. We'll talk a lot more about that in our next session. But today, let's think about some of these sequences and how specifically are they different. By the way, have you looked at the actual sequences? So which sequences are the closest? Not all of them, right? So if you go back to the original, right? So here you see a whole group of pangolin sequences, right? Which sequences are actually closest? Which two are here? If you go back to NCBI, you'll see that it's this one and this one, right? And you can, you see that they're actually, the release date, one was in 05, one was in 02. They are very different, in fact. So one is 27,000, one is 29,000, so different lengths. But the collection date is 2019, right? As opposed to all the other ones, which were in 2017, 2018. So this does make a lot of sense, that the more recent ones are actually much more related to COVID-19. Okay, so now to think about how these sequences are different, I think we need to understand a little bit better what is the actual entry. Okay, and one of the questions we have is how can we exclude the possibility of independent coevolution between bat and pangolin viruses compared to one originating from the other? Well, we're not saying that one originated from the other, and we'll talk a lot more about origins before, because we're not interested in understanding how did the pangolin get it from the bat or the bat got it from the pangolin. We talked about how bats are hosts that might contain multiple coronaviruses at once, and that's why we think they're an environment. There's also a lot of immune pressure on the virus, and so recombination and mutation happens at a faster rate there. But we, if you see right here, right, just take a quick look at how many sequences of coronaviruses you have from bats and compare it to how many sequences you have from pangolins. Did anyone take a look? Does anyone remember what might be approximately the difference in numbers of sequences of coronaviruses from pangolins, the number of genomic sequences, and the number of genomic sequences from bats. Okay, I'll tell you, it's you know, a different of thousands. There's thousands of sequences from bats. There's only probably 8 or 9 from pangolins. We know a lot less about pangolin coronaviruses than we know about bat coronaviruses. The reason is because bats are reservoirs of different viruses, and previous SARS-1 and MERS and Ebola viruses and a lot of other viruses came from bats. So there's a lot of information that we have about that. But to look at the specific differences, what I wanted to do is just briefly review how does the spike protein actually do what it does, and how might we understand those differences that we observe in the alignment that we did. Okay, so here we have an overview of how the spike protein first binds to the ACE receptor, and then through endocytosis enters into the cell. And the objective is to convert the RNA that it has inside into an mRNA that the cell will think of as its own, right? And to first create the viral polymerase so that the viral polymerase can actually start making a lot of the different portions of the protein, of the viral proteins, a lot of the viral proteins that are going to be needed to create the full virus. So the virus enters the host cell. The entry point is important, but then also all of these other steps to actually create the viruses, replicate its different portions of its RNA, all of these are going to be important for the virus to replicate and cause a specific disease. So as you remember last time, we talked about different modes of viral entry, replication, that can cause a persistent mild infection, that can cause an acute infection, and that plays a role in how it spreads as well. So spike glycoprotein, the two parts of it are actually very important for both of these aspects, to how it causes the disease symptoms and how it actually spreads throughout the body, throughout the different organs. Because as we know, that not all organs have the ACE receptor. So how does it then infect all kinds of different organs? How does it go into all of these places? And why do we see the types of severe symptoms that we're observing and not just cough, right? Because if it was localized, if it would just stay in the lungs, even in the alveoli, we would see some kind of a respiratory disease primarily, but we see a lot more severe symptoms. So how do we know what does a protein do and how does it actually fit into this process, right? So we have these nice charts. One way of knowing is reading papers, but how do people study this? People study this by looking at several different aspects. And we'll do all of these, some of it today and some of it in one of our next lectures. We can look at the amino acid sequences, right here, and align them to each other. So for example, we know sometimes a lot about a particular protein. For example, SARS-1 has been studied extensively because it happened a whole decade ago, and we now know a lot about what it actually did by conducting experiments. So we know what that protein did, and we can compare this one to that one and understand how it's different. We also can look at the different features, for example, where it was found and how it aligns to a class of functions. So proteins can be organized by different classes. We can also look at the 3D structure. The 3D structure of a protein is essentially how that sequence of amino acids folds together naturally by their physicochemical properties. They form a specific 3D structure. And that could be a global, so we can take a look at the overall structure, or we can take a look at some substructures involved here. And we can actually look at different motifs or structure features that can explain how it functions. For example, something is poking out, something is similar to the way another protein is organized. And then the final way to study this is by high throughput data. So we can actually generate experiments where we can do gene expression experiments, infect cells, see what kinds of things are expressed, and then try to understand how is that different from normal, for example, or how does that map into different known networks of different gene pathways and signaling pathways and metabolic pathways, etc. So we can, these are some of the ways that these things are studied. Okay, so the spike protein, how do we know the spike protein structure? So the spike protein structure is found on the protein data bank on PDB. And this is the structure for the protein before it changes, before it opens up, so the closed form. So let's go ahead and take a look at this together. And we'll do this in Chimera. Okay, how do we do this? The link that I posted right there in chat, you go to PDB. You can read about how this structure was created. You can also learn a lot about what the structure looks like and play around with it and actually see the different elements. So here you have the sequence. Here you have the protein that you can kind of spin around. So who knows why do we have 3 different colors here? Does anyone know what the 3 different colors? Okay, trimeric, 3 different monomers, right? So 3, not domains, right? So these are just 3 sequences or chains. Yes, not similarity yet, but these are just 3 different chains of the protein structure. So the structure is actually formed by 3 equal or exactly similar chains of amino acids. So if you take the sequence of that gene in amino acids, that just forms one of these. So you need 3 of them to make the full protein. And so the protein, as you can see, when it folds to form this structure, this shape. Okay, so here you can download the files and you can download the fastest sequence, the PDB format, right? Different formats. So what we want is the PDB format. Okay, so download this to your computer. And then if you already have Chimera on your computer, you can open it up like this. Okay, and originally when you open it up, it will look like this. Right. Okay, so if let me know if you were able to open this sequence. It might look like this, or it might look like this. So let me know if you were able to open this up. I'll just give you a couple of minutes and just maybe type yes when you were able to open it up. All right, great. Anyone else? Okay, all right, great. So if you are in Chimera X, you will have this button right here. Yes, dark background. So you can change the background to gray, black, or white. You can also do it from here, white or black. So these are just representations. I prefer the white background, but you can keep it in black. Okay. All right, so this is what we all see, right? Okay, so if you have, if you open this up just like me, you'll have a button here called sequence. So that will help you see the actual sequence, and you'll see here chains A, B, and C. You can also open up this little window right here that tells you about the 3 different models, right? So here is just, okay. I'm not sure. But so you see the three different chains, right? To help us in our next step of what we want to do, we actually want to find a specific portion of this. We can change this to a surface view. Right here you have show surface. Okay, you will also see these little molecules on the side, and these are actually sugar molecules that cover almost the whole area down here below, apart from the very top, which is an important feature here that we'll talk about in a second. But after you do this, you can just type in here in a command, color light gray, for example, or you can color it in any other way. Okay, so what you'll see is that everything just becomes kind of white, and this will help us focus on specific regions that we will identify in the sequence. Okay, so let me know when everybody got to this point. Okay, so just open the sequence, make sure that it has a surface, and color it light gray. Only the missing structure, right? So you don't need the missing structure. Make sure that right here you've got these selected. Yeah, when you first load, it actually takes some time. So you can, try this and do you see? Okay, do you see the full surface like this, or do you see something else? All right, so I'll just continue. Now here we have the sequence of our protein, right? So the question is, how can we find specific differences that we observed in the alignment that we did? I don't see the green AAs. Okay, yeah, so this coloring actually, you can right click here and you'll have settings. And it actually doesn't matter. You can select these, and this is going to show you different structural components of this, but don't worry about it right now. We're not going to use that right now. Okay, so we know that there are commands, we know that there are different views, and we have different structural features that we can select here. So just remember that, that's going to be helpful. Okay, what do we want to look at now? The spike glycoprotein has several different functional domains. So these are the signal peptide, the RBD, or the receptor binding domain, FP, which is the fusion peptide, HR, the heptad repeat, or heptad repeats, there are multiple ones, TEM, which is the transmembrane domain, and the CP, cytoplasm domain. What we see in that structure that we downloaded actually is not everything that is encoded here. Some of this had to be cleaved off to create that cryo-EM structure. So we have the full sequence of the amino acid. We don't have the full sequence which is encoded in the genome in that 3D structure. Okay, so it's just important to remember. Right here in the middle, we have the cleavage, the furin cleavage site, which is also called the polybasic site. As we'll see, it's based on a specific motif. And instead of one basic cleavage site, it became a polybasic site. So we'll see that in a second. Okay. So to understand how all of this works and how do all of these different structures fit together, there's this beautiful illustration by Falconeer Visuals that created the 3D structure kind of assembled together. And so we can see here how we have these different aspects of the spike glycoprotein, right? The spike glycoprotein structure right here, you can see the S1 and the S2, right? They actually overlap, and we'll see a little bit how. Then you see this little portion right here that has the polybasic or the basic cleavage, which is activating cleavage site, right? And then you also have this receptor binding domain, which opens up and down. So we'll take a look at that in a second. All right, so everybody, can I have the link to this pic? Yes. This is the link. Okay, so we have the top and the bottom, right? So we have the S1 subunit is this right here, which is like a cap. And then we have the S2 subunit, which actually goes inside, and this is the S2 subunit. And they are divided by this cleavage site, the furin cleavage site. So let's go to our multiple sequence analysis and find this polybasic cleavage site. So let's go back right here. Okay, and if you read the paper, you'll see that there's a specific sequence that you can actually, if you are in Eugene, you can easily find it over here. The polybasic sequence is PRRAR, right? So you can see it right here. Okay, and how does this compare between the different sequences? So you can see that only in COVID-19, we actually see this new sequence, PRRAR. Okay, so what we know is everything before this sequence is what? What is going to be before this polybasic cleavage site? Which subunit is it going to be? Right, S1, and so everything after this is going to be S2. What is in the S1 subunit? Which important part is in the S1 subunit? Exactly, so the receptor binding domain. Okay, so let's find this. So what we see here is the amino acid position on the whole genome, but to find it in the protein sequence is actually not going to be as easy as it seems. Part of it is because we don't have that sequence. It was cleaved off when that structure was created. But what we can look for is this sequence right here, QT-QTN, QT-QTN. Okay, you can see that this specific sequence, QT-QTN, is also shared with BAT-SARS-1, but not shared with, okay, sorry, it is shared with pangolin. It is not shared with BAT-SARS-1. Okay, and this is an important part for our next discussion. So let's go back to Chimera. Okay, I will just tell you from experience, you're looking for around 680, right? So here you have these positions. And remember, we're looking for QT, QTN. Okay, so tell me when you found the QT, QTN sequence. Okay, it's right there, QT, QTN. All right, great. So QT, QTN, let's select this. Okay, and now we can do color, cell. Okay, to open up this, you have to press on sequence right here. So you go to molecule display and then sequence. Okay, was everyone able to select this portion right here? Or was anyone able to find this portion? Was anyone able to find the sequence, QT-QTN? All right. All right, great. So you can select, and once you select, you just color cell red. So just type that in right here and just color it red. Okay, was anyone else able to do this? All right, yeah. No, you can't color green, only red. Just joking. Yes, you can do orange if you want, or pink, or purple, or rainbow. Okay, so what do we see? How can we characterize this little piece? Who can tell me what is special about this? What do you think is special about this little thing? What do you think is special about where this specific sequence is located from a structural aspect? Just structurally, what is different or what is special about these amino acids? Okay, anyone? Symmetric? Well, they're symmetric because these are three different chains, right? So we are actually selecting just one chain, but they show up on all three of them. What else do you think might be the reason for this? Hydrophobic, symmetric. Okay, so let's just think for a second, right? Before we don't even surface, okay, great. So we don't even want to think about the chemical properties, just physical properties. How can we characterize this? Okay, they're located between S1 and S2. They poke out, right? So if you see this silhouette, they poke out, right? Now, okay, they protrude. Great. That's a better terminology. They protrude. So if we look at this sequence, QT, QTN, and then whatever comes after, right? Let's go back to what we have here, QT, QTN, right? And then we have this insertion, PR or SPRRAR, right? And then what do we have here? Where does it continue? PSGAGS. Okay, so what we see there and here are two different sequences. In one case, we see just the sequence of the structure that is known, but here we actually see the actual amino acid. But what we know is that it's somewhere in this region and there's a missing part. So it actually protrudes even more in real life. Okay, so what is the function of this little piece? As we said, this is the site of the polybasic. Right, so this is actually the furan cleavage site. This site is a motif, right? So there's a specific sequence here that is common between many different proteins, not just this one. Yes, this 3D model is missing this part. And if you read about how this model was generated, you'll see that certain things were actually cleaved to make that model. And so you can see, right here you have proteolytic activation, herpes virus, coronavirus, flaviviruses, togaviruses. So in a lot of viruses, actually this is a site that leads to viral infections. And it actually regulates, this proteolytic cleavage regulates numerous processes in health and disease. This furan protein is a protease, and it cleaves different proteins exactly at this polybasic recognition motif. So what we're looking at is a motif. Mammalian substrates of furan include cytokines, hormones, growth factors, and receptors. Okay, so think about these cytokines. hormones, growth factors, receptors. So it's not surprising that different types of furin activity can be associated with a variety of disorders, and that includes viral infectious diseases, but also surprisingly cancer, right? Because of the growth factors and receptors. And also the enzymatic activity is exploited essentially by these viruses, right? So the virus, one of the things that it does is it uses these different sites on its proteins to cause the cell to do something that is useful for the virus. So let's think what might be useful for the virus in having this little site. So as you remember, this is a cleavage site. This cleavage site separates the S1 and the S2 domains. So if we cut off the bottom, we will actually be left with this, right? So we will be left with a unit that can do its own thing, right? So essentially think about how this protein is organized in such a way that it's simple, and yet it's organized in such a way that it can create havoc and cause all kinds of symptoms of disease as it starts spreading throughout the body. So what happens? We have that part that is cleaved, right? But before, we have the receptor binding domain, which is the S1 subunit. The receptor binding domain is responsible for connecting to the ACE2 receptor, right? And now, as you know this, you can actually find these different sections in the model that you've opened up. You probably read already about the different conserved and variable portions, right? And we looked at that conservation before. We saw that the majority of the differences is in this S1 subunit on top, right? And that's going to be obvious because of how that receptor binding domain works. How does it actually work? Well, it works by opening up. This receptor binding domain right here is a moving function. So it's a trimeric protein. Only one of the parts actually goes up to connect to the receptor binding domain, because in real life, this whole structure is also very dynamic. What is this dynamic regulated by? It's actually regulated by the process of the virus being closer to the cell membrane. So it goes next to the cell membrane, and the physicochemical properties of the different exposed amino acids actually cause it to move, so that when it is close to the ACE2 receptor, it is going to open up. And that opening up is going to be critical for it to connect to the ACE2 receptor. So this is another model also that you can find on PDB that represents the connection part of the ACE2 receptor. So the ACE2 receptor, they're separate. This is the closed version, and this is when it is open up. So the receptor binding domain is this portion, and you can see how it connects here. So when we're looking at the specific residues that are different, we're looking at the differences between the cleavage site. We're looking at the differences in the receptor binding domain as well. Other parts that are important to remember for how this whole protein functions is the heptad repeats. These heptad repeats form the fusion machinery of the virus. Once this cap is off, so the cap connects to the ACE2 receptor, falls off, and then this whole protein changes its structure to become a fusion membrane protein. So in other words, it fuses the membranes of the host cell and the virus cell. Okay, so to kind of connect all of these things together, how does this protein work? The protein connects to the receptor binding. There's cleavage of the top S1 subunit, which then is released. And remember that it's still active, right? So it's active. What does it do? That's another question. And then you have a completely separate machinery that is hidden underneath, right? So the antibodies, for example, cannot recognize any part of this fusion machinery because it is hidden by this top part. and the top part is actually exposed to interact with receptors and with the cleavage, the furin protein as well. So then this causes a transformation which connects the two membranes together. So the whole system, right, and the changes that we observe at the sequence level, right? So when we look at the sequence level and we know that this is the S1 subunit going this way, this is the S2 subunit going this way, the S1 subunit is going to be responsible for one part of the job, the S2 is going to be responsible for the second part of the job. So the question is, well, how does this actually cause disease? And what kinds of symptoms, and why are these symptoms so severe? So it turns out that the process of entering into the cell and leaving some of these pieces, some of these proteins outside circulating, actually contributes to this interferon response that causes the cytokine storm. Right? And so when we think of the entry and the replication itself at first is going to be contained, because what the virus does is actually connect the two membranes of the cells eventually. Eventually what happens is the bottom part of that spike protein is also disconnected from the virion particle, and it's also exposed in its spike form, and it's like a needle, and that needle can connect into all kinds of membranes. And what it helps the virus do is evade the immune response by connecting different cells together, which makes the virus replicate and spread between cells without being outside of the cell, and therefore not being recognized by the immune system. But at some point, all of these cells start collapsing, right? So they start breaking down because of the active virus replication. Remember where this happens. This happens in the lungs, in the alveoli, which are very thin layers of sometimes single cell layers, but maybe just a few cells in thickness. So there's not a lot of cells. It's not a tissue like skin or something like that, very delicate tissue. And if you understand the response to that, right, one of the things that it causes is the activation of the STAT1 JAK pathway, which is associated with a lot of different viral infections, but also associated with scarring of tissue. So it damages the tissue, and eventually, when the virus is released, it actually starts the cytokine storm because of the intense immune response to a lot of viral load. And so what I wanted to do for next time is actually start talking about the host response. And we prepared a data set that was published in Cell not so long ago. It's going to be focused on this response, which is directly associated with how the disease symptoms that we observe in patients. Yes, I'd be happy to share. So these are all the actually from publications that we're going through, right? So all of these are from the publications that we're going through, and the purpose of putting them here is for you to go back and to read those publications, hopefully. So you're happy, you can take them from there. And here I just want to explain kind of what we are going to try to do when we speak about the host response. So as I mentioned, the virus enters in and causes inflammation, causes the immune response, cytokine storm. The cytokine storm is related to a specific pathway that is activated that is called the JAK-STAT1 pathway. And what you see here is one of the drugs that is currently being explored as an effective method of suppressing that kind of an immune response, called ruxolitinib. And ruxolitinib is an inhibitor of this pathway. and is essentially blocks the signaling of that STAT pathway, right? The cytokines themselves. So what we'll take a look in our one of our future lectures is gene expression of different types of cells, including alveoli, lung, epithelial cells, and lung cancer cells, and see how this drug and also infection by these different viruses, SARS-CoV-2 and the respiratory virus RSV, cause different responses. What you see here is principal component analysis that we'll do together based on these three different cell populations. But before we go there, I wanted to make sure that everybody is familiar with some of these methods. So if you do not have any experience with RNA-seq processing host response, I want to make sure that all of you have enough time to at least go through a couple of courses. I saw that some of you have already started, but I want to emphasize these two first courses, Transcriptomics 1 and Transcriptomics 2. If you can take a look at those courses before our session dedicated to host response, I think it's going to be very useful. We're actually going to analyze this data. If you have enough time, go through Transcriptomics 3 and 4 because the same data set also has single cell data. And we can actually look at the single cell data, and to understand what the single cell data tells us, we need to be able to understand how it's generated and how to analyze it. So please take a look at this collection. The link is right here. And if you have any questions about those courses, I'll be happy to answer those. It will take some time. These are fairly condensed courses that have a lot of information in them. So if you can take the time to actually go through them, start now. You won't have enough time if you start, right before the session. But I'll just briefly tell you what is in those courses. So the first one is dedicated to processing raw reads, mapping, and quantification of RNA and isoform expression. And that's going to be important because we want to understand the different levels at which these genes are expressed. I don't think you need to pay for the courses. I think they're included in the program. We're going to take a look at that after the session, but I'm pretty sure they're included in the program. They should be. Okay, here you will understand the differential expression. How do we understand differences? So yeah, so okay, so Bibsha, maybe you can paste a link to courses for the program where people can find them. So in Transcriptomics 2, it talks about differential gene expression and factor regression analysis. How can we understand, statistically speaking, which genes are statistically higher expressed or lower expressed between groups of samples? And in Transcriptomics 3, we talk about machine learning, right? And so here in this study, we'll actually look at that paper and we'll see how they did differential gene expression. We'll see how they did principal component analysis. We'll see how they did network analysis to understand how these pathways are affected by infections by different viruses. So let me also give you the link to the paper. Okay, maybe I didn't put the link here, but I do have it here. Okay, there's actually going to be two papers. So this is the paper that I was talking about that has a very interesting set of data that we'll find on NCBI. We will look at how this virus causes a specific cell response, and we'll take a look at a lot of the things that we already looked at from a differential gene expression standpoint, and also from principal component analysis. So please take a look at this. I think it's open access, so you can get access to the full paper right here. The data is actually hosted on NCBI, so we'll download the data. We'll go through the process of downloading this data and preparing the analysis ourselves and getting additional insight that actually is not covered in the paper itself, which I think is going to be interesting. And we'll also review the submission of this paper to bioRxiv to understand some of the critique that was put forth about this paper before it was published. Okay, so that's what I had for today. I hope that this was somewhat informative. Again, please take a look at some of these courses. They will help you prepare for those future lectures about host response. I hope that you are starting to understand how different proteins of viruses cause disease and what symptoms are associated with them based on their process of cell entry, replication, and eventually spread in the host between multiple tissues. What we'll do next time is we'll focus exclusively on a hands-on session to study the origins of human infection with MERS, SARS, and SARS-2 pandemic. So a lot of the questions that came up in this session and the previous session, and we'll start talking about how we can come to conclusions based on sequence analysis, but also based on what we know about viral infections in different hosts, and also how they are used to, how they previously have been spread between different hosts. So let me know if you have any questions. I'll be happy to answer those now, or if you have any other questions for next time, please make sure to post those on the forum so we have enough time to prepare for the next session. All right, thank you. All right, great. If anyone has any questions, again, I recommend posting those on the forum. If there are no other questions, we're going to end the session for today. And if you cannot find any of the files that we dealt with today or have not yet had a chance to do the analysis, I'll try to put those files and the links to all of the files. And as you saw, the tools were already posted there. So please try to recreate some of the things that we did today. That will help you really get a hands-on experience and adapt that to whatever project you eventually want to do. Thank you for joining today, and looking forward to seeing you again next time.